Good afternoon. We are group 4 and then we are presenting on the catabolism of heme into bilirubin. Yeah. So basically, when normal red blood cells are produced into the circulation, they have a lifespan of 120 days. So after the 120 days, they are destroyed or broken down. And the product formed as a result of the destruction is the hemoglobin. And then this hemoglobin too, it is broken down into heme and globulin. And the heme is the iron containing part and the globulin is the protein part. And then this heme is degraded mainly in the reticuloendothelial system, which is mainly the spleen, liver, and the bone marrow. So the final product formed out of the heme catabolism is the bilirubin. So the heme part of the hemoglobin forms 80 to 85% of the main hemoglobin of the senescent red blood cells. And then after this process is complete, the remaining also forms the myoglobin and then the cytochromes. And then as I said earlier, the reticuloendothelial system forms or are the spleen, liver, and the bone marrow. And then that is where this process mainly occurs.
Destruction of red blood cells. The old red blood cells are phagocytosed by macrophages. This is a process whereby the old blood cells, which lifespan are 120 days, are destroyed by macrophages. And also after they have destroyed, they are removed from the circulation and their components, primary iron, will be formed or recycled. This hemoglobin will split into two, of which we have globin, which is a protein, and the heme, iron-containing pigments. Globin is broken into amino acids. Release of iron. Iron is removed from heme and also transported by the transferrin. When the iron are removed from the heme, the macrophages which have been break down the heme group via the enzyme heme oxygenase, then it will release ferrous iron, which is iron 2 plus. After the releasing of the ferrous ion, this ion is either stored locally within the macrophage in the form of ferritin or exported into the blood via the transporter or ferroportin.
So with the formation of bilirubin, the enzyme HAEM oxygenase utilizes oxygen and the electrons to break the bond, which produces the bilirubin, the green pigment, carbon monoxide, and iron.
Okay, so with the formation of bilirubin, when red blood cells break down, hemoglobin is released, and the heme part of hemoglobin is first converted into bilirubin. That is usually the green pigment. And the enzyme bilirubin reductase will help to convert bilirubin into bilirubin. And with this stage where bilirubin is formed, it's usually yellow in color. And at this stage too, it's insoluble in water and is usually called the unconjugated bilirubin or the indirect bilirubin. So this is how bilirubin is being formed in the bloodstream. Okay, so this slide will also explain how the bilirubin is being transported. So from the formation, the unconjugated bilirubin that is being formed is insoluble in water, and with that one, it can't be transported or move freely in the bloodstream. So it has to bind to a protein called the albumin to form a bilirubin albumin complex and is then transported into the liver. So what happens next in the liver is that the bilirubin will be converted into a conjugated bilirubin, that's the water soluble form, that can be transported or moved freely into the bloodstream. Then this will allow it to be excreted into the bowel for further metabolism.
Okay, we have hepatic uptake. Point one, we have liver cells take up bilirubin from blood. The liver cells remove bilirubin from the blood circulation, and this process helps prepare bilirubin for further metabolism and excretion. And the second point is bilirubin separates from albumin. In the blood, bilirubin is bound to albumin for transport, and at the liver, bilirubin dissociates from albumin before entering cells. And we have enters hepatocytes for conjugation. Thus, free bilirubin is taken up into hepatocytes by carrier-mediated transport. And the inside cells, it is directed towards conjugation for elimination.
Conjugation in liver. Enzyme UPD. This enzyme in the liver characterizes bilirubin conjugation. It makes bilirubin easier to process. Bilirubin combines with glycolic acid. Bilirubin combines with glycolic acid inactively. This converts into more soluble form. Forms bilirubin D-gluconazide. Two glycolic acid molecules attach to bilirubin. This forms bilirubin gluconazide. Con- this conjugated bilirubin water soluble. This product is conjugated bilirubin. This water soluble and ready for excretion.
Okay, so the next process is excretion into bowel. And the first step is conjugated bilirubin is secreted into bowel. This process takes place in the hepatocyte and its insoluble conjugated bilirubin is being converted into water soluble conjugated bilirubin. Then it travels through the bile duct. In the bile duct, the conjugated bilirubin forms part of bile, and bile comprises of salt, cholesterol, water, and other waste materials. This bile is being transported into the small intestine, where the final transformation is being done, and then it gives to its characteristic color.
I'll, I'll be talking about the intestinal conversion. Um, the intestinal conversion of heme to bilirubin is a specialized multiprocess where heme is broken down in intestine through bacterial action. Um, while some of the majority heme to bilirubin action and conversion occurs in the spleen or liver, the intestinal mucosa plays a significant role in metabolizing blood, food dietary heme or blood dietary heme. We have the intestinal bacteria which convert bilirubin into urobilinogen, and some of the converted urobilinogen is reabsorbed into the blood, and others too are converted into steatobilirubin in feces.
Okay, I'll be talking about the urinary excretion. So after the conversion of bilirubin into urobilinogen, and a small amount of urobilinogen enters the kidney through the bloodstreams. So in the kidney, urobilirubin is oxidized by the help of oxygen, which leads to the yellow pigment called the urobilin. So the urobilin is excreted in the urine, giving the yellow color of urine.
Okay, so we are continuing from the
clinical significance. Excess bilirubin leads to jaundice, as seen in hemolytic anemia, liver disease, and bile duct obstruction. Measurement of bilirubin helps diagnose liver disorders. So these are some of the clinical significance of um, bilirubin. So in summary, when aged RBC or old RBC are broken down, they release hemoglobin. The hemoglobin contains the heme part and the globin part. The globin part contains the protein or amino acid, and the heme part also contains the iron. The heme part further break down by an enzyme called the heme oxygenase, which utilizes oxygen and NADPH to produce carbon monoxide, biliverdin, and ferrous ion. So the biliverdin further breaks down into bilirubin by an enzyme called the biliverdin reductase, which reduces the biliverdin into bilirubin. The bilirubin that is produced is unconjugated, which further enters the hepatocytes to be conjugated. So in conclusion, heme breakdown is an important physiological process. Produces bilirubin through several enzymatic steps. Liver plays a key role in detoxification and excretion. Understanding this pathway aids in clinical diagnosis. Thank you.